Basics of Medical Biochemistry. First edition, 2024. Chapter 21. Vitamins, Part 3 of 4. By Professor Dr. Muhammad Jawad. MBBS, Dao, M.Phil Philosophy, Ph.D., Dip M.E. Head of the Biochemistry Department-Fazaia Ruth Pfau Medical College, Air University-PAF Base Faisal-Karachi-Pakistan. Ex-Professor of Biochemistry, Qassim University Kingdom of Saudi Arabia. Ex-Professor, Head of the Biochemistry Department and Vice Principal. Liaquat College of Medicine and Dentistry and Fatima Jinnah Dental College, Karachi, Pakistan. 21.10 Vitamin B3, Niacin. Niacin or vitamin B3 is a member of the vitamin B complex family and is also a water-soluble vitamin. Niacin, C6H5O2N, is the simplest of all the known vitamins. Chemically it is a pyridine 3-carboxylic acid or nicotinic acid. Its amide derivative is nicotinamide. It is also known as the pellagra-preventing factor, anti-black tongue factor, or vitamin G, after Goldberger's death, who discovered it in 1920. Niacin in pure form is a white crystalline substance. It is soluble in water and ethyl alcohol but is less soluble in ether and benzene. It is stable in the air and resistant to heat. It can be prepared by oxidation of nicotine in vitro. The conversion of niacin to niacinamide or nicotinamide takes place in the kidney, brain, and liver. Figure 21.13 Structure of vitamin B3, niacin or nicotinic acid and nicotinamide. Sources of vitamin B3. Good sources of vitamin B3, niacin, include yeast, meat, poultry, red fish, example tuna, salmon, cereals, legumes, and seeds. Milk, green leafy vegetables, coffee, and tea also provide appreciable amounts of niacin. Milk and eggs contain very little or no niacin but are good pellagra-preventive foods because of their high content of tryptophan. Niacin is not a true vitamin as it can be produced by the metabolism of the essential amino acid tryptophan. The liver can synthesize niacin from tryptophan. 60 mg of tryptophan can produce 1 mg of niacin. It is produced by the kynurenine anthranilate pathway. Riboflavin, vitamin B2, pyridoxine, vitamin B6, and iron are required for its synthesis. A deficiency of both vitamin B2 and vitamin B6 decreases the conversion of tryptophan into niacin. Absorption, transport, storage, and excretion of vitamin B3, it is mainly absorbed in the small intestine. At high concentrations, niacin is absorbed almost completely by passive diffusion. If niacin concentration is low, then it is absorbed by sodium-dependent, carrier-mediated, facilitated, diffusion. A small amount may be absorbed in the stomach. There is no storage of niacin. Excess of niacin is metabolized in the liver to a variety of chemicals. These metabolites are excreted in urine as N-methylnicotinamide and 2-pyridone. Recommended dietary allowance, RDA, and adequate intake daily, aid**, of vitamin B3. Infants, 2-4 mg. Children, 6 to 8 mg. Men, 12 to 16 mg. Women, 12 to 14 mg. Pregnancy, 18 mg. Lactation, 17 mg. Functions of vitamin B3. 1. As coenzyme, niacin is part of two very important coenzymes nicotinamide adenine dinucleotide, NAD+, and nicotinamide adenine dinucleotide phosphate, NADP+, which are involved in a variety of redox reactions, as a large number of enzymes belonging to the class oxidoreductase are dependent on NAD or NADP. NAD acts as a coenzyme for many dehydrogenases, about 700. It exists in two forms, oxidized, NAD+, and reduced, NADH plus H+, form. It is mainly involved in ADP-generating reactions. NADP is the phosphorylated form of NAD+. It also exists in oxidized, NADP+, and reduced, NADF plus H+, forms. It is mainly involved in biosynthetic pathways, example fatty acid and cholesterol synthesis. 2. Pellagra prevention, it prevents pellagra by keeping the redox reactions normal. 3. Cholesterol level, it is involved in lowering the blood cholesterol level. 4. CNS stimulant, it has stimulatory effects on CNS. 5. Vasodilator, it is a good vasodilator and produces a burning and itching sensation. Deficiency of vitamin B3, pellagra, the overall clinical picture resulting from a deficiency of niacin is known as pellagra, pel equals skin, agra equals rough. It is commonly referred to as 3DS as dermatitis, dementia, diarrhea, and 4th D for death if not treated properly. 1. Dermatitis, it is the inflammation of the skin characterized by dryness, thickening, and pigmentation. Changes occur mainly in the areas of the skin exposed to sunlight like the neck, dorsal part of feet, ankle, and face. 2. Dementia, it occurs due to demyelination of the spinal cord. The main symptoms include headache, insomnia, vertigo, depression, drowsiness, dizziness, and mental psychoses. 3. Diarrhea and other GI symptoms, these include anorexia, nausea, vomiting, abdominal pain with alternating constipation and diarrhea. Toxicity symptoms are due to niacin-induced prostaglandin-mediated painful flush, hives, and rash, niacin flush, excessive sweating and blurred vision. Liver damage, impaired glucose tolerance and gouty arthritis may also occur. 21.11 Vitamin B5, pantothenic acid. Vitamin B5 or pantothenic acid is another member of the vitamin B complex. It is also referred to as provitamin B5 and pantothenol. It is first isolated from yeast in 1933. It is also available in commercial form as calcium and sodium salts or as an alcohol pantothenol. Pure pantothenic acid is a viscous hygroscopic oil that is chemically unstable. Sources of vitamin B5. The name pantothenic acid is derived from the Greek pantothen meaning from everywhere, as small quantities of pantothenic acid are found in nearly every food. It is present in the highest concentrations in many organs such as the heart, brain, liver, kidney, and yeast extract. Some common dietary sources include dairy products, pulses, legumes, eggs, vegetables, cauliflower, cabbage, potatoes, and tomatoes, and whole grain cereals. Structurally, it is composed of pantoic acid joined to beta-alanine by a peptide bond. It is stable to heat but destroyed by acid and alkali. Figure 21.14 Structure of vitamin B5, pantothenic acid. Absorption, transport, storage, and excretion of vitamin B5. It is present in food either in free form or as a coenzyme. The coenzyme form is hydrolyzed to free form by intestinal pyrophosphatase. Free pantothenic acid is absorbed from the upper part of the small intestine by sodium-dependent transport system. After absorption, it enters portal circulation and is transported to various tissues. Adequate intake daily, aid, of vitamin B5. Infants, 1.7 to 1.8 mg. Children, 2 to 3 mg. Adults, 4 to 5 mg. Pregnancy, 6 mg. Lactation, 7 mg. Functions of vitamin B5. 1. Formation of coenzyme A. Pantothenic acid is an essential part of coenzyme A. As a part of coenzyme A, pantothenic acid involves in the metabolism of fats, proteins, and carbohydrates. It is essential in the formation of ADP and turns essential for energy metabolism. It is essential for heme formation in hemoglobin and essential in the synthesis of acetylcholine, which is important for nerve transmission. Pantothenic acid is first phosphorylated to form 4-phosphopantothenic acid. 4-phosphopantothenic acid is then converted into 4-phosphopantothenylcysteine by the transfer of cysteine molecule to 4-phosphopantothenic acid. 4-phosphopantothenylcysteine is decarboxylated to form 4-phosphopantethione, which is converted to D-phosphocoenzyme A by transfer of AMP. D-phosphocoenzyme A is finally phosphorylated to form coenzyme A. Figure 21.15 Structure of coenzyme A. Coenzyme A is the requirement of many enzymes as a cofactor such as a pyruvate dehydrogenase complex, alpha-ketoglutarate dehydrogenase complex, fatty acid synthase complex, thiolase, and HMG-CoA synthase. Similarly, Several molecules acting as important metabolites have coenzyme A as their structural component, like acetyl-CoA, succinyl-CoA, acetoacetyl-CoA, malonyl-CoA, etc. 2. Fatty acid synthesis. Pantothenic acid is also found in the molecule of an acyl carrier protein, ACP, which is involved in the synthesis of fatty acids. 3. Protein synthesis. According to new research, vitamin B5 is actively involved in the post-translational modification of proteins. 4. Use in cosmetic industry. Pantothenic acid is used by the cosmetic industry as a part of shampoo to provide better hair care. 5. Control of obesity. Certain studies hypothesized that pantothenic acid also has an effect on weight management and is found useful in the control of obesity. Deficiency of vitamin B5. The deficiency of pantothenic acid is rarely seen in humans. Patients who demonstrate deficiency symptoms of vitamin B5 are usually malnourished. Its deficiency is produced experimentally by giving antagonists of B5 such as omega-methyl pantothenic acid causes burning feet syndrome. It is characterized by numbness and tingling of toes, burning pain in the feet, gastrointestinal disturbances, sleep disturbances, dermatitis, and depression. As vitamin B5 contributes to sugar metabolism, its deficiency causes increased sensitivity of body cells to blood sugar levels. This causes a drop in blood glucose levels, resulting in hypoglycemia evident in weakness, restlessness, and irritability. A deficiency of pantothenic acid leads to depigmentation of the hair in rats. Atrophy of the adrenal cortex with necrosis and hemorrhage may also occur in animals including rats. 21.12 Vitamin B6, pyridoxine. Vitamin B6 is a water-soluble and heat-stable vitamin. It is a pyridine ring derivative. It consists of 3 closely related compounds, i.e., pyridoxine, alcohol, pyridoxal, aldehyde, and pyridoxamine, amine. Active forms are pyridoxal phosphate, PLP, and pyridoxamine phosphate. Figure 21.16 Structure of vitamin B6, PNP, pyridoxine/pyridoxamine phosphate, PLP, pyridoxal phosphate. Sources of vitamin B6. Plants, whole grains, legumes, cauliflower, cabbage, etc. Animals, egg yolk, liver, kidney, milk, meat, etc. Absorption, transport, storage, and excretion of vitamin B6. Absorption of vitamin B6 occurs in the proximal jejunum by passive diffusion. In the mucosal cells, all forms of pyridoxine are converted to pyridoxal. It is transported in the circulation bound to albumin. It is stored in the tissues as its coenzyme form, i.e., PLP, mainly in the liver, brain, kidney, and muscle. It is excreted as 4-pyridoxic acid in urine. Recommended dietary allowance, RDA, and adequate intake daily, aid*, of vitamin B6. Infants, 0.1-0.3 mg. Children, 0.5 to 0.6 mg. Men, 1.0 to 1.7 mg. Women, 1.0 to 1.5 mg. Pregnancy, 1.9 mg. Lactation, 2.0 mg. Functions/biochemical role of vitamin B6. Pyridoxal phosphate, PLP, pyridoxal 5-phosphate, P5P, is the active form of vitamin B6. It acts as a coenzyme in a variety of enzymatic reactions. There are more than 140 PLP-dependent activities recognized by the Enzyme Commission. Pyridoxal is converted to PLP by reacting with ADP. This reaction is catalyzed by the enzyme pyridoxal kinase. Pyridoxal plus ADP, pyridoxal phosphate plus ADP. The usefulness of PLP arises from its ability to covalently bind with the substrate, and then to act as an electrophilic catalyst, thereby stabilizing different types of carbon anionic reaction intermediates. PLP is associated with amino acid metabolism. It is involved in transamination, decarboxylation, deamination, transsulfuration, and condensation reactions. 1. Transamination. Aminotransferase or transaminase enzyme is responsible to perform transamination. Two clinically important aminotransferases are alanine aminotransferase, ALT, or serum glutamic pyruvic transaminase, SGPT, and aspartate aminotransferase, AST, or serum glutamic oxalotic transaminase, SGOT. All aminotransferases require the prosthetic group pyridoxal phosphate, PLP, which is derived from pyridoxine, vitamin B6. 2. Decarboxylation. Vitamin B6 takes an active part in various decarboxylation reactions. For example, during the decarboxylation of amino acid, CO2 is removed from the COOH group of an amino acid by the enzyme decarboxylase. Coenzyme for this reaction is pyridoxal phosphate. The decarboxylation product of amino acid is an amine. The main sites of the decarboxylation reaction are the liver, kidney, brain, and intestine. A neurotransmitter, gamma-aminobutyric acid, GABA, is produced by the decarboxylation of glutamic acid. Similarly, dihydroxyphenylalanine, DOPA, derived from tyrosine is decarboxylated to dopamine by aromatic amino acid decarboxylase. Pyridoxal phosphate, PLP, is the cofactor for this reaction. The formation of serotonin and histamine also occurs as a result of the decarboxylation of 5-hydroxytryptophan and histidine respectively. 3. Deamination. Oxidative deamination of various diamines such as cadaverine and putrescine need PLP as a coenzyme for diamine oxidase. Deamination of the hydroxyl group containing amino acids requires PLP. For example, the conversion of serine to pyruvate by the enzyme serine dehydratase. 4. Transsulfuration. PLP plays an important role in the metabolism of sulfur-containing amino acids. Transsulfuration from homocysteine to serine occurs in the synthesis of cysteine. It is catalyzed by the PLP-dependent enzyme cystathionine synthase. 5. Heme synthesis. In mitochondria, succinyl-CoA combines with glycine in the presence of PLP to produce delta-aminolevulinic acid, ALA, which is an intermediate in heme synthesis. It is the rate-limiting step in the biosynthesis of heme. 6. Fatty acid synthesis. It catalyzes the biosynthesis of fatty acids such as arachidonic acid from linolenic acid. 7. Glycogen synthesis. It appears as an integral part of glycogen phosphorylase concerned with glycogen metabolism. 8. Metabolism of threonine. Threonine aldolase is the enzyme that can convert threonine to glycine. This enzyme needs PLP as a coenzyme. 9. Metabolism of tryptophan. It acts as a coenzyme in the conversion of tryptophan to niacin. It is produced by the kynurenine anthranolate pathway. Riboflavin, vitamin B2, pyridoxine, vitamin B6, and iron are required for its synthesis. A deficiency of vitamin B6 leads to the formation of xanthurenic acid. 7. Amino acid and K+ transport. It is involved in the intracellular transportation of amino acid and potassium. Deficiency of vitamin B6, decreased dietary intake, alcoholism, and impaired absorption are the main causes. Some drugs act as anti-vitamin B6 such as chronic administration isoniazid and penicillamine. The main clinical features are hypochromic, microcytic anemia, lymphocytopenia and leukemia, indigestion and convulsion, growth retardation, dermatitis and other pellagra-like symptoms, peripheral neuritis, depression and mental confusion, the appearance of xanthurenic acid in urine. Toxicity of vitamin B6. Large dose of vitamin B6 can cause severe nerve damage. More than 200 mg per day cause peripheral neuropathy. Doses higher than 500 mg per day may be toxic, leading to serious central nervous system problems such as pain in limbs, numbness, clumsiness, imbalance, and difficulty in walking. Further high doses of vitamin B6 can increase the severity of cadmium-induced anemia and also cause peripheral neuropathy. Therapeutic uses of vitamin B6. Helpful in the treatment of vitamin B6 responsive sideroblastic anemia. Useful to prevent urinary stone formation. Cure vitamin B6 deficiency seizures in infants. 21.13 Vitamin B7, biotin. Biotin is water-soluble B-complex vitamin formerly known as anti-egg white injury factor or vitamin H. It mainly participates in carboxylation reactions. Biotin is a heterocyclic sulfur-containing monocarboxylic acid. It is derived from imidazole. It is formed by the fusion of imidazole and thiophene rings with a valeric acid side chain. Figure 21.17 Structure of vitamin B7, biotin. Biocidin is a coenzyme form of biotin. In biocidin, biotin is covalently bound to the epsilon amino group of lysine. Sources of vitamin B7, eggs, liver, kidney, yeast, pulses, nuts, and vegetables are rich sources. Biotin is also present in cereals and dairy products to some extent. Adequate intake daily, 8, of vitamin B7. Infants, 5 to 6 g. Children, 8 to 12 g. Adults, 20 to 30 g. Pregnancy, 30 g. Lactation, 35 g. Functions of biotin. Biotin is required for carboxylation reactions, i.e. CO2 fixation reaction. It acts as a coenzyme for several carboxylases such as pyruvate carboxylase, acetyl-CoA carboxylase, propionyl-CoA carboxylase, beta-methylcrotonil-CoA carboxylase, etc. There are some carboxylation reactions in the biological system which are not dependent on biotin. For example, the formation of carbamoyl phosphate in the urea cycle and the incorporation of CO2 in purine synthesis. Deficiency of biotin. Biotin deficiency is not commonly seen in humans because of its wide distribution in foods as well as the synthesis of this vitamin by the bacterial flora in the gut. The main clinical features of biotin deficiency include severe dermatitis, weakness, nausea, and vomiting along with muscle weakness. Dermatitis is associated with the loss of hair around the eyes. Biotin antagonists. Avidin is a heat-label protein present in raw egg white. It can bind to biotin, present in egg yolk, making it unavailable for absorption. That's why avidin is also known as the raw egg white injury factor. Avidin is inactivated by boiling the eggs and thus biotin is readily absorbed when boiled eggs are consumed in the diet. A total of 4 biotin molecules can bind to a single avidin molecule. Intake of 20 raw eggs per day can produce biotin deficiency in humans. Prolonged use of antibacterial drugs such as sulfonamides can also cause biotin deficiency. Laboratory use of biotin. Many lab tests use biotin technology due to its ability to bond with specific proteins which can be measured to detect certain health conditions. For example, biotin is used in hormone tests and tests for markers of cardiac health like troponin. The affinity of avidin to biotin is greater than most of the usual antigen-antibody reactions. Avidin-biotin system is commonly utilized for the detection of pathogenesis in ELISA tests. DNA is generally labeled by radioactive nucleotides. At present, biotin labeling of DNA is employed more commonly. Biotin is added to nucleotides, which is then incorporated into the newly synthesized DNA. The fixed biotin can be identified by reaction with avidin. End of Part 3 of 4 of Chapter 21. Thank you.